Welcome to this tutorial for multi-arm, multi-stage designs in nQuery. In this tutorial, we will cover how to do a multi-arm, multi-stage group sequential design sample size calculation in nQuery by looking at an extension of the enhanced trial for COPD. Multi-arm, multi-stage designs are an increasingly common adaptive design, as the emergence of master protocols and the associated platform and basket trials have led to a large increase in trials which allow us to evaluate multiple different treatments within the same study. With the multi-arm, multi-stage framework, we have the opportunity to remove and add arms flexibly depending on if an arm is being proposed to be promising or has been proposed to be lacking promise based on interim analysis or on other data. The multi-arm, multi-stage group sequential design is an extension of the traditional group sequential framework for greater than 2 groups, where we use a Dunnett-like test for the multiple comparisons. The Dunnett's test is a test which compares each treatment group or dose level to the control group, and there is no direct comparison between the treatment groups. In practical terms, when designing a MAMS group sequential design, we are evaluating the power and sample size based on the dose level or treatment group which gives the maximum effect. However, the power based on the maximum effect does not exclude the opportunity to drop individual arms that are found to be futile at an interim analysis. And this can be done without affecting the overall type 1 error. However, dropping an arm may decrease the power somewhat, but since the arms are likely to be highly ineffective at an interim analysis, if considered for dropping, the effect is relatively minor on the power as well. As the group sequential framework is well understood in the context of clinical trials, this extension to the MAMS context can be seen as an easy way to introduce the concept of MAMS design that is consistent with the good operating characteristics of group sequential design. In nQuery, the sample size calculation for a MAMS design consists of 2 parts. In the main table, we need to specify our parameters for the overall study, such as the number of arms and the number of looks, and our expected significance level and power. And we also need to specify the spending functions that will be used for the efficacy and/or futility bounds. In this case, the researchers wanted 0.025 significance level, with the number of arms equal to 3. Note that this is the number of treatment arms with an additional placebo or control arm being added to this for a total number of arms of k plus 1, or in this case 4. The number of looks in this case we'll set to 2. Remember that this includes the final analysis, so this is equivalent to 1 interim analysis and 1 final analysis. As this method is based on some simulation, we may wish to enter a seed here to ensure consistent results if we attempt to replicate the same example, and there was a target power of 90%. For the spending functions, in this case they had an O'Brien-Fleming efficacy bound, which is the default, though there are other options available from the alpha spending function dropdown, and they wanted a non-binding O'Brien-Fleming beta spending function. Non-binding indicates that even if an arm were to cross the futility bound, you can still choose to continue the study even if that happens. Remember that in the context of a MAMS group sequential design, the efficacy and futility bounds are in terms of the maximum effect achieved, and only if the maximum effect were to cross the efficacy bound or be below the futility bound would we stop early. However, as mentioned previously, if one arm were to be performing poorly, you have the opportunity to drop that arm without affecting the overall type 1 error control. In this particular case, they looked at the effect sizes in terms of difference from control. And thus the first row here will be taken as being the control arm with a mean response of 0. Then the mean responses in the 3 dose levels considered in the enhanced trial were assumed to be 0.14, 0.16, and 0.18, increasing as dose level increased in this case. And the standard deviation was assumed to be the same in each group of 0.5. And so we enter 0.5 in each row here. Note that the default allocation ratio is that there will be equal sample size per group, and since that was used here, we'll retain the default here. The MAMS procedure iterates over a lot of different scenarios and therefore will take a little bit of time. But in the interim, we can look at some additional information here. You can see to the right-hand side of the mean response table, there is the group sequential output table. And this will include the sample sizes expected for the control group, as well as the efficacy and futility bounds, as well as the nominal and beta alpha, nominal alpha and nominal beta, and the incremental and cumulative alpha spent. These use the same types of procedures as explained for a standard group sequential design. So, based on the inputs we gave, it requires a total sample size of 640, or 160 per group. You can see here that if the standard z statistic, which was the maximum, was greater than 3.272, we would stop early for efficacy. In other words, if at least one of the arms was this successful, we would stop early. However, if the maximum effect was below 0.853, we would stop early for futility. In other words, if all of our arms were below this threshold, we would stop early for futility. Although, given it's a non-binding bound, we would have the option to continue, although it would not be recommended. We can then see that at the final analysis, if the maximum effect is above 2.358, we would find the results significant, and that at least one group, and indeed the maximum group, would be effective, and if it was below 2.358, we would find the opposite and find for non-significance. You can see that we spent 0.02002 alpha at the first look and around 0.02 of our beta at the first look as well. And you can see that these alphas are effectively the alphas that you would need if you're using a standard Dunnett's test on the interim data. So if the Q of the significance level of a standard Dunnett's test was less than 0.001 based on the interim data, then we would stop the trial early for efficacy. Obviously, this can be easily extended to multiple arms and multiple looks. However, as you increase the number of arms and looks, the calculation time will increase as well. And so we have provided the 3-look design and 4-look design that are equivalent in this table here. You can see that the sample size has increased from around 640 to 668 for an equivalent 3-look design and 684 for an equivalent 4-look design. The side table on the left is the same, but you can see that as the number of looks increases, the number of interim analyses and columns in the GST table has increased proportionally as well. So as mentioned, if we were to drop the 0.14 response arm at any interim analysis, the overall type 1 error of 0.025 would still be controlled under the Dunnett test methodology, but the power may be decreased slightly. However, since this arm is expected to not be the best arm, it's unlikely that that power decrease will be very significant. To learn more, visit statsolves.com. Thank you for watching.